Hi friends, you are here with me, Professor Girish Kukreja, back again. In the earlier video, we talked about the flow chart of the tests which are to be performed to come to a conclusion that which particular sugar is present in my particular given sample. So today we'll be talking about how to perform these tests. One of the first preliminary tests when we went for identification of the sugars was your Molischs test. So a general test which is answered by which is answered by all carbohydrates. So some of the general tests we'll be discussing today, that is your Molischs test and the anthrone test, answered by all the carbohydrates. So whether your given sample it contains monosaccharide, disaccharide or polysaccharide, this test will be given positive by all. So if you want to find out that whether your given sample it contains any of these carbohydrates or any of the carbohydrates, you will go for the Molischs test or the anthrone test. Now talking about both these tests, the general principle basically remains the same. Both of these tests, they contain concentrated sulfuric acid, maybe the Molischs test or the anthrone test. So in presence of this particular acid, all the glycosidic bonds, they are hydrolyzed and you release what is called as your monosaccharide, the monomeric units of the carbohydrates. These monosaccharide, again in the presence of the acid, are dehydrated to form the hydroxymethyl furfural, or what we in general call as the furfural derivatives. So these furfural derivatives then react with the reagents which are present in the respective test. So talking about the Molischs test, so what will happen in the Molischs test is the same, that in presence of the concentrated acid, the glycosidic bonds would be broken. Once these glycosidic bonds are broken, monosaccharides are released, they would be dehydrated to form what is called as your furfural derivatives. These products, they will then react with the sulfonated alpha naphthol, which is another reagent in the Molischs test, to form a purple colored complex. So if you get this purple colored complex, you can be sure that your sample contains any sort of what is called as your carbohydrate. So basically what you will require to perform this Molischs test is you will require what is called as your 5% alpha naphthol in ethanol. So 5% weight by volume of your alpha naphthol in ethanol. So how you will prepare, like you will take 100 ml of your ethanol, in that you will add 5 gram, or more precisely like you will take some 90 ml of your ethanol, add 5 gram of your alpha naphthol and then take the volume to 100 ml. So you are going to require a few drops of it. So if you have what you call as very less amount of test solution to be tested, you can like reduce accordingly. Like for 50 ml you will use around 2.5 grams of your alpha naphthol. So yes, so this is your alpha naphthol reagent, and yes, you are going to require concentrated sulfuric acid. Now talking about performing this test, so to perform this test you will take around 1 to 2 ml of your test solution. In that you will add this 2-3 drops of your alpha naphthol reagent. One precaution needed to be taken here is that prepare this particular reagent fresh, because this alpha naphthol is quite unstable. So whenever you want to perform this test, see that you prepare this alpha naphthol reagent freshly. So add 2-3 drops of this freshly prepared alpha naphthol reagent, and now again carefully from the sides of the tube, right, from the sides of the tube you will add say 1.5 ml of your concentrated sulfuric acid, and then you will find that the two layers they get separated, and at the junction of the two layers, if at all any kind of carbohydrate is present, you will get the formation of the purple or purplish brown colored ring. So appearance of this brown purplish colored ring indicates that the test is positive. As I told, this is answered by all carbohydrates, even your polysaccharide, your glycoproteins, they will also answer this kind of what you call as your Molischs test. Another particular test which we generally do is your anthrone test. For this you will require 0.2% of your anthrone reagent in concentrated sulfuric acid itself. So you will take say around 100 ml of your sulfuric acid, in that you will add this 0.2 grams of your what you call as your anthrone reagent. The principle remains the same, that in presence of the concentrated sulfuric acid, the sugars they are hydrolyzed, the glycosidic bonds they are hydrolyzed, the polysaccharides they are hydrolyzed, they form monosaccharides which are then dehydrated to form the furfural derivatives, which then react now here with the anthrone reagent to form a bluish green colored complex. So again, you take around 1 ml of your test solution. To that you will add around 1.5 to 2 ml of your anthrone reagent. You will keep it for 5 minutes. Probably it will not require heating, but then still, if you don't get a bluish green color, to just confirm, you can go for heating in the boiling water bath for around 5 minutes or so. Formation of this bluish green colored complex will indicate that the test is positive. So formation of a purple color ring at the junction of the two layers in case of the Molischs test, and appearance of this bluish green color in case of this anthrone test will lead you to the conclusion that some sort of carbohydrates are present in your given sample. Stay tuned with me, Professor Girish Kukreja, for more specific tests on carbohydrates, proteins, amino acids, lipids and much more. Thank you.